Hi everybody, and welcome to the Fascinate session on DNA extraction from strawberries. My name is Dr. Jill Johnson. I am a senior lecturer in biosciences at Aston University in Birmingham, and I work a lot with DNA. So today we're going to learn how you get DNA out of cells. So hopefully everyone has their copy of the protocol, DNA extraction from strawberries. That was part of the Fascinate package, so hopefully everyone can take a look at this as we're going through this process. So the first thing we need to look at is the list of materials that we need. So the first thing we're going to need is a plastic bag. So any kind of Ziploc plastic bag will do. You obviously need a strawberry, which we have right here. And you need a couple of different liquids to be able to make this protocol work. So the first liquid you need is DNA dissociation buffer. Now this buffer is mostly tap water, and it also has in it some washing up liquid and some table salt. So the washing up liquid helps break open the cells from the strawberry, and the table salt helps solubilize the DNA, and it keeps it in the water phase until we're ready to take it out of that water phase, and it also helps separate it from the proteins that are also in the cells. You're also going to need some materials to filter your strawberry. So we're going to have a liquid phase that we want to collect. So I've just got a simple funnel from the lab and a little bit of cheesecloth as my filter. You could also use a coffee filter for this, or if you wanted, you could use a piece of fabric or a bit of tights just over a glass tumbler to filter your strawberry dissociation liquid into. We also need some ice cold alcohol. So I'm using 100% ethanol. So this is a reagent that we use in the lab. So I've got this here. If you're doing this at home, obviously you wouldn't have 100% ethanol. You can use strong drinking alcohol, such as vodka or gin or rum, but it needs to be very high proof, and you will need adult supervision if that's what you're going to be using for this process. Alternatively, you can use surgical spirits that you can just pick up at the chemist, but whatever alcohol you use, please put it in the freezer for a couple hours before you do your DNA extraction, because the alcohol works best when it's cold. We also finally will need a long wooden skewer of some kind. You can use a barbecue skewer. I've got a coffee stir stick here. A wooden spoon or a chopstick would also work. You just need something that has a rough surface that the DNA can stick onto at the very end. All right, so that's what we need in terms of materials, and we are now ready to go. So the first thing you need to do is to take the green bit off of your strawberry, because we don't want that. Those cells are pretty hard to break open, and the fruity bit of the strawberry is a lot more useful for this purpose. So you just want to pop your strawberry into that bag, and then gently squash the strawberry into kind of a jelly. So try to get it as mashed up as you possibly can, just using your fingers, just really gently make some strawberry goo out of your strawberry. So what we're doing here is essentially increasing the surface area of all of those strawberry cells so that our dissociation buffer can more easily get in there. Now, you can actually do this with any kind of fruit. Strawberries are really good for this because they're what we call octoploid, which means every strawberry cell has 8 copies of all of its DNA, whereas in humans, we only have 2 copies of all of our DNA in our cells. So as you can see, there's a lot more DNA in a strawberry cell than in a human cell by weight. All right, so the first thing we're going to do after we've squashed our strawberry is to add our dissociation buffer. So if you're making your dissociation buffer at home, you'll just want to fill a glass with tap water, add 1 teaspoon of washing up liquid, and 1/2 teaspoon of table salt, and that is really all you need to do for dissociation buffer. So I'm using a popular tool we use in the lab called a serological pipette and a pipette dispenser, and I'm just going to add 10 milliliters of dissociation buffer into my bag, and then I'm gently going to mix that up. And you definitely want to be gentle here. You want to avoid generating too many bubbles, because bubbles mean that you have less liquid to work with at the end. And everything you want is now in this liquid stage. So that's what it looks like at this point. We've got our mashed up strawberry with our 10, I think I put in about 13 mils of washing the dissociation buffer in there. So yeah, just enough to make sure you've got your strawberry mash sort of well saturated with that liquid. Now this is a little bit of a tricky part here, and it could generate a bit of a mess. So if you need adult supervision and someone to help you, make sure you've got someone to help you around. Otherwise, you might need to clean up a little bit of a mess at the end. Now, if you wanted to, you could use a pair of scissors and cut the end of this baggie off. I'm just going to try to carefully pour my strawberry liquid through my filter and my funnel into this tube that I have here. And of course, you can just do this into a drinking glass at home. So just give everything a bit of time to pass through that filter and just collect as much of that liquid from inside your bag as you can. Oh, see, I'm making a little bit of a mess. It's a messy protocol. All right, so I'm just going to make sure I have all of my liquid out of my bag, because that's where the DNA is, and that's the bit that we want to collect in our tube. All right, I think I've got about as much liquid as I can reasonably get here. All right, so I'll put that in the bin later. Now what we have is some liquid containing dissociated strawberry cells. So that will have DNA suspended in that liquid. Remember, the salt is helping to keep that DNA in the aqueous, the water solution. So the last thing we're going to do now is to add our cold ethanol. So again, I'm going to use a serological pipette. I'm going to add about 10 milliliters of ethanol, about the same amount of liquid as the water phase. And right away you can see that the DNA comes out of the bottom phase and moves into the top phase where that ethanol is. And now all you need to do is take your wooden swizzle stick and you can lift that DNA out of the tube and have a play around with it. So yeah, you just need a dry bit of a wooden stick and you can get a good bit of strawberry DNA out of that tube. Now, because we've used fairly safe materials, as you can see, I have gloves on because I'm used to wearing gloves in the lab. But if you're not wearing gloves at home, it is safe for you to have this on your hands. But obviously you don't want to put this in your mouth because there's washing up liquid in there and that doesn't taste very nice. So just remember, this is not for eating, but you can definitely play around with it. So there you go. You have now some DNA from strawberries. I hope you enjoyed this session, and I hope you share it with all of your friends and tell them about how you can extract DNA from strawberries and have a go at home with this particular exercise. Have a great day. Bye.